Baert. I'm a project manager with Genome Prairie. I'm the project manager of the SeekNet project. And today we've got 3 speakers. They're going to talk about PacBio. I'd like to start by acknowledging that Manitoba is on the traditional lands of the Anishinaabe, the Cree, the Oji Cree, the Dakota, and the Dene, as well as the birthplace of the Métis Nation. I'm coming to you from Treaty 1 territory, and we respect the treaties and we're very grateful for the use of the land. So first I'd like to show a few slides about SeekNet, then we're going to get right into all of the speakers. And here we are. Oops. So I just wanted to let you know about the SeekNet project. It's funded by the Canadian Agricultural Partnership, Manitoba Agriculture and federal government, as well as Genome Prairie. And it has the goal of increasing adoption of genomics technology in the agricultural sector in Manitoba. So if you want more information, you can contact me if there is anything that you're interested in learning about any further, or if you want to learn about collaborations or just meet other people within the genomics community in Manitoba, please contact me. And I'll stop sharing my screen. So I'm going to send things over immediately to Alex.
Yeah, and I'll just start sharing right away here. Just got a quick slideshow to go through. So thanks everyone. I'm going to just jump right in because we are running a bit behind and we've got a lot to get through. We've got this scheduled for an hour, although I think it might be an hour and a half on the actual invite. We're going to try to fit it in under an hour with some questions. If it goes over, we'll stick around if people want to. If they don't, we can obviously sign off as need be. So just a couple housekeeping notes. Like I said, if you want to submit any questions, just put them in the chat and we will address those at the end. If we don't get enough time to go through them all, we're happy to address those one-on-one with you at another time. We are recording today and it will be available after, and there will be a brief survey at the end, which is obviously optional, but would be great if you could take it. So these are who's speaking today. I'm on the left here, Territory Account Manager for Western Canada. I cover from Manitoba West. I'm based in Vancouver. Sean Walkowiak is a research scientist at the Canadian Grain Commission, and he is very familiar with PacBio hi-fi reads as he's been using them in his lab for about a year now or so. And Jacob Brandenburg is a market development lead with PacBio, and he'll be talking a bit more on the specifics of applications with hi-fi reads, whereas I'm going to just do a quick technology overview. So that's exactly what I'll jump into, just a really basic overview of hi-fi sequencing. So as you can see, there's at the top here, the applications of areas that we work very well in: human genomics, microbial genomics, plant and animal, cancer research, gene therapy. So we're not limited to plant and animal, but we're going to be focusing on that for this call. If anyone on here is from other application areas, you might learn a little bit anyway by sticking around, particularly with the basics of hi-fi here. And if you're more interested in getting detailed information or discussing one-on-one, we're happy to do that. So feel free to reach out. Our website also has a wealth of information. This top banner is actually from our website, so you can go there and learn more and reach out to us if you want to learn more. As far as other what you can do with hi-fi reads, obviously assembly is one of our really great features. Because of the high quality data, assemblies are really quick and easy. Epigenetics, we're the only one that can call direct off the sequencer 5MC. Variant detection, which works great with the high accuracy we have. Targeted sequencing is an option. RNA sequencing, which we call iso-seq as well. So you'll hear those terms used interchangeably. And complex populations. So a lot of people that have tried hi-fi reads really notice the difference. So it's one of those situations where you don't really know what you're missing until you've tried it. You can't see it unless you actually see it, so to speak. And if you're using technologies that don't allow you to see the difference, you won't notice it. You think you're getting everything you can. And what we find is that people that try it, as you see by just some of these quotes, we get a lot of these all the time, but once people try hi-fi, they're really sold on it and they really notice a big difference between other technologies. I liken it a bit to something like a high definition TV where, for years you watch your standard definition TV, you've got this really expensive TV, you think it looks great, and then suddenly high def comes out and you realize how bad your standard definition was, and now it's actually painful to watch on standard definition TV. Sort of same sort of idea once you try hi-fi. So what makes it so much better and makes everyone so excited? In the old paradigm, you had the choice of short reads that were a very short read length but highly accurate, or you could do long reads, which is our old technology, and ONT still has this type of technology, which is long reads. You get really long reads, but you compromise on accuracy. So you really aren't getting a very accurate read, and you're kind of having to compromise between the two technologies. When we developed hi-fi reads about 3 years ago, it really changed the game and it separated us from the competition. The idea being that now you can get a 15 to 25 KB read length at highly accurate scores, which are actually even higher than short reads. So there's no need to compromise anymore, no need to do both technologies. I'm not saying that short reads don't have a place, they certainly do. There's always going to be a place for both technologies, but if you're looking for sort of a single technology to get the most data out of it in a very high quality fashion, hi-fi reads are the way to go. So what's the underlying technology of hi-fi reads? Well, you start with a double-stranded DNA. We add these ligate adapters, which is smart bell, we call smart bells. That allows the primers to anneal and bind to the DNA polymerase, which then creates a circular DNA template. And what happens is it sequences it in repeat phases, and the polymerase reads and trims are trimmed to the adapters to the subreads. So you get these subreads, and what this allows is sort of a proofreading. So our errors are random, meaning that they won't necessarily occur at the same time every pass. So you can wash out those random errors and only get true errors. Other systems use more of a systemic or systematic error process, and that's when it's actually in the systematic technology, you're actually not able to wash those reads out. So once the system's done this sort of proofreading, it then calls a consensus read, which we call a circular consensus read from those subreads, and that's your hi-fi read, which is 99.9% accurate and up to 25 kV. So this just gives you a visual on the left here of the sort of length of the reads. This is drawn to scale. This is a 200 base pair Illumina read, and you can see here how much longer 20,000 base pair PacBio hi-fi read is. And that's really, the thing to note here is that the accuracy is actually better with this long read. So, you can imagine how much easier it is to work with that kind of data. And on the right here, when you're looking at, this was a study done by the FDA called the FDA Truth Challenge, Precision Truth Challenge. This was an independent study. We had no influence on it. None of the manufacturers did. They looked at the 3 technologies listed here. And what you'll notice is PacBio, again, we had the highest accuracy, not a lot higher than short read, but higher. And what's most notable, though, is not only do we have significantly higher than ONT, but the range of our reads are very tight. So really, you're getting at least 99%, and most of them are falling in this 99.9 something percent range. Whereas if you're using something like ONT, you can get some reads that are fairly high, and down here they intersect with us. But the majority of your reads are actually going to be quite low, and that creates a lot of unknowns and difficulty working with that kind of data because it's just not consistently high. And even when it is high, it's not nearly as high as we are. So it gives you a good idea. And this actually, to sort of focus on the chart here, this is the actual raw numbers from that study where Illumina had 6.3 times more errors and ONT had 21.1. That's for SNVs, indels, and structural variants. This is just a graphic to show how long the reads can get. This is an actual read of over 19,000 base pairs. You can see this whole read is the little red dots are the 8 errors that were found. So this had a 99.96% accurate score. And you can see how long a read like this is to get right with only 8 minor errors, and it is pretty impressive. So when other papers, this is a bit more recent, this Nature Biotechnology did a performance assessment of the systems here on the left. So they looked at all the main systems and their conclusions, some of the comments on the right here, as you can see. PacBio provides the lowest error rate out of all technologies, has the highest reference rate-based mapping and lowest non-mapping rate, showed the lowest mismatch rate, and achieved the highest precision. So again, people who try this technology and compare it really notice the difference. Our awareness has been low over the years, and a lot of people don't know about PacBio, and that's really our limitation. Once people become aware, there really aren't any limitations to them seeing how good it is. This is just another couple studies that looked at, on the top here, the human genome and a couple of human genes. And with PacBio, hi-fi reads at 22-fold compared to nanopore at 47-fold, or even nanopore at 30-fold with short read polishing. So combining short reads with it to polish up the errors. And you can see here again, with one single technology at only 22-fold sequencing, you've got a much higher accuracy score, as well as a much higher complete rest-seq gene score. So, you're spending more money, more time using both technologies when you can just use one. Same with the rice genes at the bottom here. Hi-fi at 20 versus nanopore at 62-fold and short read polishing. And again, a much higher classical complete gene score, as well as far fewer frame shifted genes. So this was a really eye-opening and interesting study that was done a couple of years ago. And it looked at assemblies, and the assembly of the California redwood gene, which is this huge 27 gigabase hexaploid, very complex gene. So a lot of work to put something like this together and a lot of data to handle. They looked at ONT with 23-fold data, or 23-fold sequencing, and short read at 122-fold, compared to just hi-fi reads at only 22-fold. And you can see that just using hi-fi reads, you got a much higher assembly score that allowed you for haplotype resolution, at a much higher contig N50 score for more continuous assembly. Average alignment score was much higher for more complete genes. And the real eye-opening here was the assembly time. I mean, when you're working with such high quality data, you can reduce that assembly time from 5 to 6 months to 6 days. And this is something I constantly hear from bioinformaticians that working with this data, that assemblies that took them weeks before, they can do in a coffee afternoon on Sunday. So it really shows you how clean that data is, and you get it right the first time. You don't have to clean it up. This was another kind of groundbreaking study that showed, or at least, I guess, announcement that showed that hi-fi reads really were a game changer. Everyone knows that the Human Genome Project that was sequenced about 20 years ago. Well, they didn't actually sequence all 3 billion genes. It was about 92%. There was about 8% they couldn't do, or base pairs, I should say. They ended up unable to do it, not because they didn't want to or they didn't think it was information that was needed. They simply didn't have the technology to do it. And that was not until last year, using hi-fi reads, and it's actually specifically called out in this paper, that this 8% was not overlooked due to lack of importance, but rather due to technological limitations. And this high accuracy long-read sequencing finally removed the technological barrier, enabling comprehensive studies of genomic variation. So again, opening up a lot more information that you just couldn't see before. And just in summary, the bottom line, I guess this is sort of a graphic visual, short reads. I think a lot of people kind of know the difference here, but you're chopping it up into very small fragments, and then you're trying to put it back together. Well, that makes it very complicated. You have mismatches. You're trying to put that book back together, and you might have a word that has a missing letter there and here, and then you place it in the wrong spot, and the whole story changes. Or you're just missing information. So you end up with a book, and you're just sort of getting bits and pieces. With long-read technology, you're chopping it into much larger segments, which are much easier to assemble and put together and look at and understand. And only until what used to be the limitation of long read is, sure, you'd have these full pages, but then the words would be all blurry, right? Because they weren't accurate. So, but with hi-fi reads, you actually get a very clear visual. So you're able to put that book back together the way it should be and read it the way it should be. So again, just showing you a visual of sort of how much more information you get from hi-fi. Whoops. So just want to call out a couple things which you're interested in getting hi-fi sequencing done. Opal or GIFs, they use the names interchangeably, is a lab in Saskatoon at the University of Saskatchewan. They actually have a PacBio hi-fi sequencer. They're actually the only provider of complete PacBio services in Western Canada. They have a lot of other services, so they're full service pay for service core lab. If you're interested in getting a project done, you can go to their website at opal.gifts.ca, or you can also email partner with us at gifts.ca to talk about their project, and they'll do any kind of free consultation before receiving a quote and be able to do their service. They've been doing a great job there. So feel free to reach out to them or reach out to me if you need to set something up too. I can put you in touch. And then of course, Sean is on the call as well. Sean Walkowiak from Canadian Grain Commission. And he is not a real true fee-for-service core lab, so not the same as GIFTS, but he is available for collaborations on a per project basis. And if you want to contact him and discuss some sort of project or collaboration you have, he certainly could look at doing that. So those are your two options in Western Canada. Like I said, GIFTS being the only sort of true core lab. That's it for me. So I guess I'll pass it on to whomever is up next. Get out of this now. Everyone's gone small on me. There we go. All right.
I think that's passes it over to me. So thanks, Alex. Can you guys see the slides okay, Alex, and hear me okay?
Yes, I just muted myself. I guess I can.
I'm Sean Walkowiak. I'm a research scientist with the Canadian Grain Commission, and I'm going to be talking about the journey that took me to PacBio, and then some of our recent experiences with the technology. So the Canadian Grain Commission is a department within the federal government of Canada, and we establish and maintain standards for grain quality and safety. And we do this through our inspection division, which works closely at the ports and with elevators. And we also have a grain research lab where we have scientists that work on individual crops, as well as different technologies to support the grain sector. So in terms of crop research programs, these are sort of listed at the bottom here. So all grain crops are represented, or official grains. And we also have technology research programs. We have a grain biotechnology program that looks at GMO testing and monitoring of crop varieties, especially as part of export requirements. We have a chemistry team that trace organics and trace elements. So they do testing of mycotoxins and pesticide residues and heavy metals and those types of things. And then there's my program, which is the microbiology and grain genomics program. And we do monitoring of different microbes that appear on grain. So these would be plant pathogens as well as human pathogens, anything that's a risk to production or food safety that appear on grain. And we also do grain genomics research to support market development and the monitoring of varieties. So across all of the programs that we have in the Grain Research Laboratory, we do monitoring of cargoes as part of our exports. We monitor samples directly from harvest as part of a program called the Harvest Sample Program, which is a voluntary program that producers participate in, and we evaluate about 15,000 samples a year through that. We also do requests for service analyses. So we do have a lot of fee-for-service type activities within the Grain Research Laboratory. Sequencing is currently not set up as one, but if there's a need to support the industry, then we're there to help. So feel free to reach out. And we also, like Alex mentioned, do projects on a collaborative basis as well. And the labs at the Grain Research Lab also do plant breeder evaluations. So when new varieties get registered for Canadian producers, they need to go through a stringent testing and validation process where we assess them for quality and their performance. And a lot of that testing is done by our scientists at the Grain Research Laboratory with the Grain Commission. So when we talk about genomics, a lot of you are probably familiar with genomics on the call, but those of you that are less familiar, all things have DNA. DNA codes for RNA, which are genes, and these form proteins, which are functional units within cells and tissues. And that's sort of what gives everything its life. And DNA can vary, across individuals of the same species, as well as between different species. And that variation gives different RNA and different proteins. And that's how we get different traits and diversity amongst, species and individuals. So understanding the genomics of individuals and species is really important because it allows us to assess the diversity of different germplasm, especially in breeding, that might be present in our native germplasm within our country, or as well as international germplasm, which we could use to bring in new variation. So it allows us to also assess unrelated species, and that variation is important for allowing us to identify what's different between different crop varieties. And this sort of level of understanding at a high level is also important at an applied level when you're trying to integrate it into breeding. So the upstream and downstream are very intertwined. At a more specific level, when you're looking at the genomes, you can identify genes and traits that might be important for incorporating in breeding or for monitoring as part of variety monitoring. And these can be used in selection programs for genomic selection or marker-assisted selection in breeding. And then it's also important to assess and validate those genes in terms of their function and how they're working within the biological system. So genomics is really integrated into the understanding of the biological system and validating of genes across different cultivars and species. So over the last few decades, the cost of sequencing has gone down significantly. So back in the day, it used to cost around $100 million to sequence a human genome. Now you can get one for $200 to $600, depending on the approach that you use. But in addition to the reduction in cost, as Alex mentioned, there's differences in the technologies in terms of read lengths that allow you to really capture the genomic information of more complex genome. So when I started working with sequencing back in the early to late 2000s, I was mostly working with microbes because the reads were so short that that's all you could really work with. Doing short read sequencing of larger, more complex genomes just would not result in a genome assembly that was usable for most applications. And if you look at the size of genomes, you have bacteria, which are around 5 million bases. And if you compare that to the bread wheat genome, which is just this Goliath monster of a genome of 15 billion base pairs, it's 3,000 times larger than bacteria. Not only that, its genome is much more complex. So bacterial genome is just a single chromosome. It's a circle. It's really small. So each region in the genome is not represented more than once. It's very simple. The wheat genome has 3 different subgenomes, the A, B, and D subgenomes, 7 chromosomes per subgenome. And each chromosome within the subgenome groups are very similar. So for example, chromosome 1A would be very, very similar to chromosome 1B. So when you're getting short sequencing reads, you really have no idea which subgenome it belongs to. You need longer sequencing reads so you get enough variant information across the read to be able to properly assemble it and assign it to a genome, because it's really too similar between the subgenomes, especially of complex large genomes that are polyploid like wheat. So if I give you a brief timeline on the sequencing of the wheat genome, in 2014, we had a survey sequence of wheat, which is based on short reads and some Sanger reads. But, and I apologize for offending anybody, but I find this as genome assembly was, a major breakthrough at the time, but was pretty much unusable for most applications that I would be interested in, just because it was so fragmented. With the advent of some new library preparation techniques and assembly approaches, we were able to get a chromosome assembly in 2018. And this was a major breakthrough. This was a game changer because we could actually get access to gene information, structural variation, and that sort of thing. And then in 2020, I was part of the group that did the pan genome as part of the 10+ wheat genomes project, where we assembled the genome of many different wheat cultivars, and that was also very valuable. But at that point, we started to understand some of the limitations of the sequencing approaches that we were using. A lot of the really repetitive things and copy number variation were not being properly pulled out or represented in these genomes because of the short read technologies that we were using. So this is when Curtis Posniak and Harmeet, Harmeet's on the call here, so a quick shout out to Harmeet. we started working on generating long read data to support the next generation of assemblies that would have some of this more complex information like repeats resolved. So we've used the Oxford nanopore technologies, which require a lot of read polishing because the reads are not as accurate as PacBio reads. But the reads can be much longer, so that, it's just a different type of technology. And then supporting it with some special library techniques and for short reads and some assembly approaches to link regions that are from different chunks called Hi-C. And then we sort of did this in parallel with PacBio hi-fi data as well. So this is what we saw. So this is for CDC Landmark, which is one of the genomes of the 10-week genomes project. So this first assembly on the far left is the one that was based on short read technologies, different library prep methods. Then we have our assembly sort of in the middle here with the Oxford nanopore, and then we have the assembly on the right here with PacBio. So you can see PacBio had the fewest number of contigs, it had larger contigs, the assembly was larger, it had more of the genome represented, and the N50 value, which is like the assembly is in a chunk of this size or greater, was the largest for the PacBio assembly. And L50 is the number of chunks that made up at least 50% of the genome. So this was also the fewest. So we had fewer, much larger chunks comprising the genome. So this made it a lot easier to place the chunks into their proper chromosomes. We saw that there was a lot better resolution of genes with copy number variation. So when we started to see this with the PacBio, it became clear that PacBio is going to be an important tool in our toolbox, especially for these larger complex genomes. So in 2022, we decided to purchase a PacBio instrument with the Canadian Grain Commission. So this is a picture of the instrument here. I apologize for the mess in the lab. I had to boot some of the technicians out to take a quick picture for the presentation. So here's the instrument here on the left. And this is our molecular lab. We have a whole bunch of genotyping equipment and some Illumina sequencers and nanopore sequencers. So we're very well-equipped state-of-the-art genomics facility at the CGC. And then we started to do some sequencing of different crops that are important to Western Canadian growers. So here's the team at the Grain Commission. We went to Brandon as part of their field tours, and we got to see the oat field trials, which are headed by Dr. Kirby Nielsen at AFC Brandon. And when we were talking with Kirby, he had a whole bunch of different traits that he's interested in to develop newer, better varieties for Western Canadian producers. And as part of his pipeline, he uses genomic selection, but he really needed some really good reference genomes for Western Canadian oat varieties. So when talking with Kirby, we decided to sequence two oat varieties. One is AC Morgan, which is one of the more popular oat varieties in Western Canada. I think it's second for insured acreage. So it has a lot of those really high priority traits sorted out. And then we also sequenced Leggett, which is another really interesting variety because it has some valuable disease resistance genes, especially for crown rust. So we visited his field in the summertime. Shortly thereafter, he sent us some DNA. And last Wednesday is when we finished sequencing one of the varieties, Leggett. And we actually are still sequencing Morgan right now. It's actually on the sequencer. We're just finished, I think. And this is some of the data from one of our latest runs. So this run had 37 gigabase pairs of output. The reads were nice and long. We had a read N50 of over 20 kilobase pairs. The reads are also very accurate. The median accuracy was Q34, which is a logarithmic scale, but it basically equates to 99.95% accuracy. So the reads are long, they're accurate. And then we're also able to call some of those epigenetic modifications like methylation of 5-methylcytosine on the fly directly off of the sequencer. So this is one of our good runs from last week. This one came off, I think, last Friday from Morgan. But we actually finished sequencing Leggett on Wednesday. I think I passed off the data to Harmeet on Thursday. And then Friday, Harmeet gave me an assembly. So it was honestly that quick. It was maybe 2.5 weeks of sequencing. And then an assembly took maybe a day and a bit. And here's some of the stats from the oat assembly. So on the top left here, this is the reference genome of oat that got released in 2020, earlier this year. And this is the stats for its assembly. So it's 10.96 gigabase pairs. So our assembly is comparable in size, but look at the N50 value. So their assembly was 17.73 megabase pairs for their N50, and ours blew it out of the water. So we crushed the latest oat reference genome significantly. And we did that in 2.5 weeks. So we were really, really happy and impressed with this result of how well the PacBio data was able to assemble that oat assembly for Leggett. And hopefully it does similar with Morgan once we're done sequencing it next week. And then in parallel, we've also been working on the durum wheat pan genome as part of an international durum sequencing project. So on the top right here is the reference genome currently for durum wheat. This one's a little bit older, it's from 2019. So again, we're able to get a slightly larger assembly, so more of the genomes represented, albeit it's a different line than the one that's in the reference. But look at the N50 values. So like these assemblies are of meeting or exceeding the current standards for reference genomes. And both of these assemblies were based on the short read method that we used for the original wheat genome. So I anticipate that as more and more people integrate PacBio into their workflows, we're going to be seeing these significant improvements to the assemblies, especially for these complex genomes. So what we have going on right now, while we're sequencing other grain crops, we're using the PacBio also to call variants and compare genomes to support a variety of identification activities within the Grain Commission. Supporting breeders and their crop improvement. I mentioned our projects with Canadian Oats. We also have projects with Genome Canada, the 4D wheat project. Mentioned the tetraploid wheat plant genome or the durum pan genome, and more to come. I should say we're just getting started. Our instrument was installed in April, I think, of 2022. So we've only had it for a few months, and we're already, well on our way on the crop side. On the microbe side, we do a lot of work, as I mentioned, with plant diseases, as well as microbes that could be foodborne pathogens or cause risk to humans and animals. So we're doing some metagenomic sequencing. So we're doing amplicon sequencing. We have our first run for that plan tomorrow. And then we're also planning to do some shotgun metagenomics, where we're doing whole genome sequencing of complex microbial communities. So I'll leave that to the acknowledgements, the team at the Grain Commission and the lab that do all the real work. I'm just sort of a face to all the hard work that everybody else does. And I especially want to acknowledge Harmeet, who's with the University of Manitoba, who's the expert on genome assembly, who generated our oat and durum assemblies and the long read assemblies of Landmark. Curtis Prosniak, who's led the 10 wheat genomes project and is the durum wheat pan genome, as well as the Landmark work with the long reads, and Kirby Nilsson, who's the oat breeder out in Brandon, who's working with us on the latest oat genome assemblies. And of course, all the institutions and funders that support the work that we do. So thanks, and I think I'll pass it off now to Jacob.
Great, thank you, Sean. Let me get everything pulled up here. Okay, perfect. Are you able to see my slides okay?
Yeah, looks great.
Awesome. So yeah, my name is Jacob Brandenburg. I'm the Global Marketing Manager for Plant and Animal Sciences here at PacBio. Today, I want to talk a bit about the applications we have in the plant and animal space and give you some customer examples, hopefully that relate to your research. And I'm going to highlight the oats project as well that Sean brought up. So it's great to see that in the last couple of years, hi-fi reads are still able to improve upon those assembly metrics. Let's see if I can get this to advance. Perfect. So here at PacBio, we separate plant and animal sciences into 3 categories. So we have agriculture, conservation, and ecology. Most of my talk today is going to focus on the agricultural section. And we have an overall theme of genomics in action, so sequencing for a better, healthier planet to making sure we have a sustainable world for future generations. Here's kind of a high-level view of the areas that we support in agriculture. So people utilize our technology for breeding programs, for a lot of pest and disease work, and then more on the newer cutting-edge side of genome engineering applications. For conservations, PacBio is the main technology for a lot of the global biodiversity efforts. And our main uses here is going to be capturing biodiversity, using for species monitoring over time, and then also on the plant animal biology side. And then finally, under ecology, we're used heavily for a lot of the large metagenomic efforts, such as bioscan that are going on globally, and also metagenomics, which I know Sean alluded to a bit, but we are the leading technology when it comes to generating high quality mags as well for metagenomic assemblies. And here's a look at the biodiversity efforts that are going on globally that PacBio is the main technology for. And you're probably noticing a lot of the big names that you're used to hearing about. So the Earth Biogenome Project, the VGP, ERGA, and the African Biogenome Project. And so before I get into applications, I did want to highlight a great blog series that came out this year. So this was a 3-part series that we did based on 3 publications from the same group. And they looked at how the contiguity and other assembly metrics have changed over the last 20 years with the changing of sequencing technologies. And so if you look at where the 3 pink arrows are pointing, you can see that once long reads came onto the market, it drastically improved the assembly metrics in every area for reference assembly. And PacBio has a long historical standing with the plant and animal community. And the reason for that is building reference genomes. And as we know, there are many challenges that come along with sequencing plant and animal genomes. And they can really be broken up into 3 categories. So size and complexity of these organisms vary greatly. And as we know, a lot of these can have extremely high ploidy levels. They also have a lot of repetitive content. You know, if we look at maize and wheat, for instance, they have greater than 60 and greater than 80% repetitive elements. And then again, each project is going to range drastically in size, ploidy, and heterozygosity. So a lot of times we have to employ custom strategies, especially on the extraction side, to make sure that we're meeting the needs of the project. And hi-fi reads are uniquely suited to address these challenges because they span a lot of these repetitive elements in the long read space, but they're also highly accurate and come from a single molecule. So that allows you to phase your assemblies into haplotypes instead of getting a collapsed assembly. And here's that same paper that Sean just showed. So this is the oat genome that is publicly available currently. And this was released at PAG the last time it was held in person. And the takeaways from this is hi-fi was really just being adopted on the market. So they were able to fully annotate a hexaploid oat reference genome, assemble it into the 21 pseudochromosomes. This is publicly available. And there's now also an annotated version 2 on the grain genes. And the chromosome rearrangements are now delineated. So hopefully this approach will help readers overcome a lot of the barriers. And we're also realizing that reference genomes do not account for all the diversity that exists species-wide. So we also have been using hi-fi reads to generate pangenomes. And so you can think of pangenomes as a reference for an entire species as opposed to a single individual. And to generate these pangenomes, you sequence multiple individuals. And when you assemble those together, you start seeing what core genes of a species and what are dispensable genes of a species. And so those core genes are going to be slow to evolve over time and house the critical functions. And dispensable genes and unique genes are going to have those adapted traits and rapidly evolve generation to generation. And pangenomes are important because they can help exploit the genetic diversity of wild relatives. It's going to really help facilitate a better understanding of those agrogenomics traits and assist with breeding strategies. And again, the reason for this is if we have just a single reference, that's not going to account for all the genetic diversity that makes up the species, which is largely driven by structural variations. And so we have to incorporate multiple unique individuals to generate that pangenome. And to generate these, we recommend that you start by picking 20 to 30 genetically diverse representatives of a species. You're then going to sequence those with high quality, highly annotated genomes. And then if you want to use low pass or resequencing efforts to characterize the rest of the population, you're able to do that at this point. And so again, hi-fi sequencing can be used for all of these. And here's the applications that you would use for that. So to sequence the genomes, you're going to need to use our whole genome sequencing applications to generate those assemblies, and then use our isoseq method for that transcription level annotation. And then finally, for characterizing the rest of the population, you can do low pass sequencing to characterize all the structural variations that may exist in the rest of your germplasm. And here are some examples coming out in the publication space. So this one is a group that generated a maize pangenome. So they used 26 genetically diverse individuals, and they were able to see a lot of structural variations that they were able to then link back to phenotypic traits which were not previously able to be explained. So a great use case for pangenomes. And a similar experiment on the soybean side, they also used 26 genetically diverse individuals. That must be some kind of magic number. And they were able to identify numerous variations as well that could not be detected with short read. And you can see in the graph here the different core genes and the different pangenes that were associated with this experiment. And now that we've talked a bit about reference assembly and pangenomes, I did want to just briefly touch on pest and disease research or crop protection as well. So it's great to be able to sequence your species of interest, but it's also going to be really important to be able to assess different organisms that interact with that species. So we have different applications in this space as well. So RNA-seq for understanding those plant host adaptations, whole genome sequencing for those invasive species, and we have different workflows depending on the genome size of the organisms. We are happy to announce that we partnered with Twist Biosciences, and we now have a targeted sequencing application in the long-read capture space for finding those resistance genes and doing panels and exome sequencing. And then finally, metagenomics to really look for antimicrobial resistance genes and also interactions with soil microbiomes. And long reads do tend to take a little bit of input. So we recommend 3 to 5 micrograms for our traditional library prep workflows. A lot of times that is not possible for smaller bodied organisms. So we're also happy to have different solutions in this space. So we also have a low DNA input workflow and an ultra low DNA input workflow to allow customers and researchers to sequence single insect individuals instead of having to pool multiple together and then tease out those haplotypes in the assembly. And so here's kind of a snapshot of those 3 workflows. So the standard hi-fi workflow needs around 3 micrograms, and that really doesn't have any genome limitation size. Low input is really targeting what can be done on a single SMRT cell. So we have a 300 nanogram input limitation for that. And this is really targeting genomes that are less than 1 gigabase in size. And then finally, we have our ultra low DNA input, which needs 5 to 20 nanograms worth of starting material. This does have some long range amplification. So keep that in mind that it is going to have some of the biases that come along with PCR. And this is for targeting genome sizes that are 500 megabases and lower. And no genome is too big for hi-fi. So another great publication series and blog post that has come out is from USDA recently. They sequenced 6 different locust or desert locusts, and they were able to assemble this 8.8 gigabase genome in just 12 chromosomes. And this is just a really great experiment. All this data is publicly available and just another exciting public data set for advancing research in this space. Another area I wanted to touch quickly on today is gene editing. So we're starting to see an uptick in customers using our technology to do different gene editing applications. And the reason for this is hi-fi reads, again, are uniquely suited to give you the best visibility into multiple parts of this process. So you can use hi-fi reads to generate the high quality reference genomes that are needed to design these editing experiments. You're also going to be able to have that visibility and look at edits that are in highly heterozygous and GC-rich regions of the genome, which you cannot do with alternative technologies. The read lengths tend to be long enough that they're going to be able to span the entire edited region. And you're also going to be able to evaluate off-target effects to make sure there are no unintended consequences. And again, touching on some publications that have recently come out in this space. So here is a great one where they used PacBio hi-fi reads with common wheat cultivar. And they were able to assemble the primary contigs with the hi-fi reads with very high levels of contiguity. And they paired this with OmniSeq to be able to assemble the chromosomes into those pseudomolecules when it was possible. And because of the quality of this assembly, they were able to target specific haplotypes for their gene editing experiments, which also allowed them to be able to better monitor the different regions of the genomes that might be susceptible to off-target events. So really great publication for gene editing with even complex organisms like wheat. Another one in the gene editing space is from cassava genomes. And so this one, the researchers were using PacBio hi-fi reads more on an amplicon basis. And so they were comparing two different lines of cassava here. The first one, if you can see the red dashes, they are deletions that were found at the amplicons at this target site. And the insertions are the red font that you can see. And if we look at the bottom, you can see the different frame shift mutations indicated by those little red arrows that were picked up by the hi-fi amplicon approach. So for gene editing workflows, which is becoming an important tool for scientists globally, PacBio hi-fi reads can really help you accelerate the breeding process, which is one of the main advantages of gene editing in the first place. It's also going to be able to help you better target the genes that are going to improve those traits that you're interested in. It's going to help plants improve their different biotic and abiotic stress resistances. And it's also going to be able to just give you a better understanding of the underlying plant architecture. And the last application I wanted to talk with you about today, and this one's just a bit more exciting for us because it's a newer application, is the epigenetics. And so we're now happy to announce that we can sequence the genome and the epigenome at the same time with a single library prep on one sequencing run. And the reads are still going to be able to do phasing, have uniform coverage. And due to the nature of hi-fi reads, this allows you to have access to a larger portion of the genome to do that epigenetic calling than standard bisulfite sequencing. And so I'm sure, as we're all aware, epigenome definitely contributes to phenotype. And so these different chemical additions to the nucleotides come along with most natural organisms. And the three main categories are 6-MA, 5-MC, and 4-MC. In the past, we have been able to look at 6-MA and 4-MC for different applications, where 5-MC has been a little bit more out of reach. So happy to finally be able to bring that into the PacBio application umbrella. And if we look at how this compares to the other main approaches on the market today to get it the same, you can see that most of the approaches are short read based. They use complicated workflows that you have to do in parallel with sequencing workflows, and long read sequencing allows you to do direct detection of these methylation patterns. And so if we look at that side to side, standard bisulfite, 2 library preps, fragmented, high GC bias, and you're going to get coverage gaps. If we look at ONT, you do get some of the advantages of long read, but it's still going to have lower read accuracy, and you have to do complex analyses to get at this information. Where hi-fi reads right now are the only solution where you're going to be able to get that uniform coverage and coverage more of the genome, but you don't have to do any additional analysis. This analysis is going to come directly off the sequencer itself. And the reason we're able to do this is because our sequencing polymerase sequences native DNA as opposed to PCR product. So we're not stripping away the methylation that is naturally occurring on your DNA template. And when our polymerase hits a specific methylation pattern, the kinetic signature stalls out a little bit, and we're able to measure that time in the polymerase incorporation stall to specific methylation patterns. And so on the bioinformatics or the machine learning side, we're able to kind of generate model data sets and train models to be able to detect these specific epigenetic modifications. And this is what that looks like. So we use a pileup summary by position and by haplotype. So you can see here how the different hi-fi reads stack up. So we're able to collapse that down to a % methylated by position, but then you can also see the different hi-fi reads themselves and how those calls are being made. And it's also important to understand the coverage needs. And so we downsampled the hi-fi data coverage all the way to 5x. And at 5x coverage, we're still able to see a very high correlation to standard bisulfite sequencing. So this is also exciting for us when it comes to low pass applications. You're now going to be able to still maintain the integrity of a lot of that epigenetic information, even at very low coverage amounts. And so now when we look at hi-fi reads, we're going to be able to get the most accurate variant calling on the market for all variant types. You're going to be able to phase your genomes into their different haplotypes, and you're also going to be able to get these epigenetic modifications by haplotype as well. And so this is a human example, but just wanted to show a couple real world examples here. So if you look at the arrows at the bottom pointing to these blue sections, we're able to detect hypomethylation at these transcript start sites in human. And if we look at that same example in chicken, you can see that we're able to pick up that same hypomethylation pattern at these transcription start sites. And an example of this in the plant space was at AGBT Ag this year. So Mike Schatz gave a really good proof point on comparing the hi-fi the hi-fi CPG methylation caller to standard bisulfite sequencing in tomato and in maize. And what they were able to find was that the problem they had with bisulfite is there's a lot of these transposable elements that exist genome-wide, and the short read data wasn't able to span these regions. But when they were able to apply hi-fi reads to the same portion of the genome, where the red arrows are indicating here, you can see that these track back to different transposable elements that exist. And so EM-seq is where the red arrows are pointing to. And the hi-fi strand at the top, you can see spans this entire region, and they're able to get those CPG calls even over these transposable elements. So Mike Schatz was very encouraged by this data, confirming that it has very high correlation to standard bisulfite sequencing, but now they're able to assess portions of the genome that were unobtainable by the previous methods. And so where do we go from here? As we know in plants, CPG is really only one methylation or one of the main contexts for 5MC, but we're also interested in looking at CHG and CHH. So kind of just wanting to give a little bit of an update on the map here. So we're hopeful that we'll be able to apply this same methodology to some of these patterns as well. So if we take a look at CHG, this is also going to be a symmetrical methylation type. So it's going to exist on both strands. So as our hi-fi polymerase is making multiple passes around a single molecule, it's going to be able to read that methylation pattern every time around. So we're confident that we'll be able to train our models to pick up this methylation pattern. We just need to find the right truth sets. CHH is going to be a little bit more challenging because that methylation pattern only exists on one strand. So we may need to have additional coverage to be able to get the level of detection for CHH that we can for CG. But confident that this is still something that we can add to the PacBio roadmap at some point. And then one last application in the epigenetic space I did want to highlight is chromatin fiber sequencing. So this is coming out of a group from the University of Washington. They have developed a single molecule chromatin fiber sequencing methodology that they're happy to share if you ever want to be put in contact with them. And this helps you assess the chromatin structure of the underlying DNA, essentially. And so if you're interested in learning more about this type of approach, feel free to check out the publications in the space, or please connect with Alex and myself, and happy to get you more information on this type of approach. So what are the possibilities of methylation data, and how can this be used in the plant space to kind of further a lot of these programs? We know that methylation has an effect on gene editing efficiency, which has not been well categorized. So hopefully now that we're able to get to this level of information, hopefully this will make targeting different portions of the genome with gene editing even more efficient. Assessing DNA methylation for transformation events in plants. So again, we know that methylation plays a role in how genes function and also how they get passed on to multiple generations. So if we transform a specific gene into a plant of interest, but it doesn't have the appropriate methylation patterns, how many generations will that gene stay in the plant before falling out? So experiments to assess that are needed. We're now able to do tissue-specific methylation experiments as well. And so that may be another area of interest to researchers in this space. Assessing the chromatin structures and its effect on gene regulation, which we went through a bit with FiberSeq. We're also seeing evidence that alleles that appear homozygous at the DNA level are heterozygous at the epigenetic level. So this may allow us to phase additional alleles more so than just looking at the DNA information alone. And then finally, we're now going to be able to assess larger, more repetitive portions of the genome with this type of detection. And so, in summary, PacBio right now is the only instrument on the market that you can get the PacBio or the whole genome sequencing information complete with phasing and the 5MC information on instrument. And we're happy to add epigenetics into our total package offering for hi-fi reads. And so with that, I'm happy to take any questions you might have. I know that was a little bit rapid fire, but also thank you for your attention and inviting me to speak here today. So Alex, I'll send it back to you for the Q&A portion.
Yeah, all right. So I'm just going to quickly put up my screen here. Just a couple of, whoops, all the way back here now, sorry, go through all this again. So just a couple notes on what's coming soon. MOS-Seq kit is coming soon, as well as these twist enrichment panels that Jake mentioned. We also do have a short read, highly accurate short read sequencer called that uses a technology called sequencing by binding that's coming out too soon. So we're going to be the only company that has both high accuracy for long and short reads using native DNA. So that's really exciting. Now questions. So there's a scan here for the QR code if you want to take a brief survey. That takes you to SurveyMonkey. If you want to contact the scientist after this, you can go to pacb.com/scientist or for future events. And then that's my email at the bottom. I have to close this screen off so I can get to the questions, I think. So we did have a few questions coming through the chat.
Alex, I don't think you're sharing your screen, so people can't.
Oh, did I not share that? Oh, sorry. I thought I did. Oh, okay. I'm going to put that up again then quickly. Sorry. So yeah, I guess I didn't do that because it's where's share. Where did you guys go here? And sorry, guys. I just got to get this back to share screen. There. Now it should share. Is it sharing now?
Yes, we can see
Yeah,
it now.
we can see it, but it's not in presentation mode.
Okay, so that was the I went back and did this again. Sorry, I should have done this slide. All right, so this was the MOS-Seq and the twist enrichment I was talking about. Here's this survey. So if you can scan this, there's your survey, and I will leave in the emails my email at the bottom as well as that. So sorry about that, everyone. But what I will do is I will leave this on the screen, but because I can actually see the questions here, so that's okay. I can leave that up. So while I do this, I'll read a couple of questions. First one came from Miranda with Dana Gamage, and it said, regarding microbial sequencing with PacBio, why would you choose PacBio instead of other sequencing platforms? And why would you choose PacBio for shotgun metagenome sequencing? I'll let Jacob address that.
Yeah, happy to take that. So on the microbial sequencing front, PacBio is going to give you the best assemblies possible. So a lot of times you're able to assemble your microbial sequences into a single contig, so a circularized assembly. And on the shotgun metagenomic side, PacBio technology is actually going to give you the most high quality mags per gigabase spent. So it's going to be the most cost effective on that front. If you're looking at it from a high quality mag standpoint, you're going to be able to get like species level identification on the 16S side and be able to pull out the 16S region from the shotgun metagenomics. And you're also going to be able to get more like genes per hi-fi read for shotgun metagenomics type research. And you're also going to be able to assemble a lot more organisms from your metagenomic sample into like complete assemblies.
Great. So the next question from an anonymous attendee: has PacBio been used to identify pest disease resistant studies?
Yeah,
Maybe
so
I can answer that one really quick.
yeah, Sean,
I
please feel
think,
free to take that.
I actually have a study open from wheat where they assembled the genome of an African wheat variety, and they used PacBio to do that and clone the gene for yellow rust resistance YR27. So I think especially with disease resistance genes and complex genomes, there's a lot of copy number variation, especially in those receptors. So the long reads are really important for getting at that to be able to properly characterize those genes.
Great. Next one from Vinaru Bersuring. Have you used PacBio on plant microbiomes?
Shauna, have you done any experiments on that specifically? Otherwise, I'm happy to take that.
I will be tomorrow. We have our first run going, but you can take that one because we haven't generated any data with that yet, but it's in the works. Perfect.
So I would say that I can definitely pull some experiments or some publications on plant microbiomes if you're interested. But definitely we have a lot of other communities as well that you can look at. So we have everything from water to soil and like wastewater, et cetera. So lots of examples of different microbiomes, and confident that we can do plant microbiomes on PacBio.
Great. We've got one more written question, but I've got Harmeet here with his hand raised, so maybe I'll let him ask a verbal question first, if that's the idea.
Yeah, thank you so much. So the question is like regarding the methylation analysis. Is PacBio offering any workflows to integrate? So probably the methylation analysis is attached to the reads when they come off the equipment. The IGV plots you were showing, does PacBio has a workflow solution to it, or is it like in the public domain, we have to find our tools ourselves?
Great question. So the instrument is going to store those calls in the BAM files, and you can visualize those calls in IGV. So I guess you still are going to have to like use visualizations and assemblers to get at that data itself specifically, but that information is all stored in the BAM file itself, and you can visualize it easily with a lot of the programs that you're familiar with already.
Okay, yeah, thanks.
All right, we've got one from Jerlene Halliday. How long are the reads that are generated from the IsoSeq platform pipeline? And are they long enough to identify and quantify gene isoforms?
Yeah, so I'm happy to take this one as well. And I did not spend much time talking about IsoSeq. So for IsoSeq at PacBio to not get filtered out during post-processing, we have to be able to see the poly A tail, the 5' and the 3' end. So what we're left with is only full-length isoforms. So essentially, the limitation here is what we're able to do on the cDNA generation side. So we've seen isoforms as long as 10 to 15 kb, and they're definitely long enough to identify and quantify gene isoforms. That's one of the advantages for using IsoSeq over alternative methods.
All right, we got another couple ones coming in here, so good conversation. Sylvie Cluche asked Jacob specifically, could you say something about your new targeted method works?
Yeah, happy to. So we partnered with Twist Biosciences. So Twist is going to be in charge of probe design, and they have optimized the capture and different polymerases to work better in higher GC environments and for longer fragments. So that protocol is really targeting 7 to 9 kb regions, and it's hopefully going to be able to work better for GC content than other capture methods on the market. A lot of the work was initially done in the human space for developing dark region panels. And so we are still looking for more people in the plant and animal space to help us test out this protocol and make sure that it's still going to work for the plant and animal community, because Twist is able to change how those probes are tiled across your regions of interest. So if you do have applications in mind, please reach out. Be happy to talk more about you and see if there's any type of pilot projects that would be a good fit for this protocol.
We got another one from Geraldine. Is it possible to use PacBio to do dual RNA-seq on infected plant tissues to identify pathogen vectors or virulence factors and plant-resistant genes simultaneously?
We would have to sit down and take a look at exactly what you're proposing. So the one caveat to the PacBio isoseq method is that our standard method uses the poly A tail to do the first strand synthesis. So if you're looking at different RNAs that exist genome-wide from both the infected plant tissues or the pathogens, you would just need to make sure that they're polyadenylated. And if they're not polyadenylated, or only some of them are polyadenylated, we may have to discuss custom approaches. So it is possible to use a different first strand primer, but now we're kind of talking about targeted approaches as well in the RNA-seq space. So if you have a use case in mind, it's best to reach out to your local PacBio team or the core that you're using to do the sequencing, and just make sure that we don't need to do some type of custom approach beyond what the standard isoseq protocol is capable of.
Yeah, I think that's it for questions. So I don't know if Racquel, you want to sort of sign off. I'm just going to thank everybody. And like I said, please feel free to reach out, or, this will be, as Racquel said, posted for recording too. So you can share that or watch it later, but I'll let Racquel say the final goodbyes.
Yes, I just wanted to thank all of you for speaking, and of course, our audience for participating and listening closely. We will have it posted on our website for those of you who missed it. So thank you to everyone.
Thank you.
Thanks, everyone.
Take care.